So I'm standing before you a little bit emotional now. I just want to say a heartfelt thank you to Jim for giving such astonishing talk. It's quite a hard act to follow at the best of times, but today I felt it pulled my heartstring a little bit more. Jim doesn't know this, but in 2012, when my son was newborn, I was at a summer school in Cambridge, one of the first Lena Peltonen schools. And Jim was a tutor there, and I brought my son, and Jim said two things to me. I don't know if you remember, Jim. The first thing you said is that keep on collaborating and sharing data, and everything will be fine. And I followed that almost slavishly throughout my career. And I'm very pleased with that. And the second thing Jim said to me was that throughout your career, you will be tempted to sort of immerse yourself into your science and forget yourself. But look at your husband and your child and remember that your family life outside of your science is as important for your creativity as your science is. I don't know if you could call this. I'm a happy mother of 3 children today, and I'm still happily married to the same man. So thank you, Jim, for that. I will, however, not talk more about my family today, but I will talk about my scientific family. So we'll see if I can push forward. The first, I have a disclosure. I'm a part-time employee of PHP, and my husband is an employee and shareholder at Vertex Pharmaceuticals. None of the companies have any influence of the work I'm talking about today. So this is my scientific family. These are people that have dedicated their time, their excellence, their smartness, and their scientific endeavor into what is our lab's success. So I am accepting this award on behalf of everyone who is on that slide. I tallied the number of co-authors I've accrued over my career so far, and I tallied 673. I might have forgotten someone, and I do apologize, but there are too many to name here individually. I will, however, say, as was presented, that I worked in the bosom of a range of consortia, but particularly the giant consortia, which I feel I want to give a particular shout out in this setting. So I've sort of pledged a big part of my career to tackling obesity. As we heard, it's one of the most persistent health problems in our society today. It's one of those traits where a lot of people feel an ownership. Everyone has a personal relationship with obesity and their own body shape, and therefore a lot of people feel that they're experts of the traits. Obesity is a multifaceted heterogeneous condition, a disease as defined by WHO, and we most commonly measure it by BMI over 30 in adults, in men and women. We should remember, and I will come back to that later in the talk, that obesity actually shows signatures of sexual dimorphism, a distinct difference between men and women. There are two paradoxical states that are often getting discussed and a little bit misinterpreted in the field, and it's the healthy obese. It's when you have obese body shape as defined by BMI, but you have a low fat percentage, thinking about athletes, for instance, but also the slim obese, where you have a normal BMI or low BMI even, but a high fat content. And as you will see here on the right, looking at UK Biobank work by Nick Baya and Duncan Palmer, you will see that in about 20% of people, BMI isn't closely correlated with the fat content in the body. Oh, wrong. So I've been really inspired by Ronald Kahn's work. So he and Stefan Gesta published a bunch of theories about how when you start to accumulate fat over the body, you go from a lean body into an obese body. There are two sort of generalized patterns that obesity can take, where the first one is a pear shape or gynoid obesity. It's also female obesity, which is correlated with a low risk of diabetes and metabolic syndrome. Keith Freiner and Frederick Karp in Oxford have done beautiful physiological work showing that it can even be protective of some of the cardiometabolic traits. The other sort of facet of this is when you start to accumulate fat visually and you have an android or male-shaped obesity where you have a high risk of sort of cardiometabolic traits. If we look at the genetic underpinnings of this, we know from overall obesity, beautiful work from Sadaf Faruqi, Stephen O'Reilly, and other people in the field, that if you have single gene mutations, you will have extreme phenotypes of hyperphagia, which is overeating, and extreme early onset obesity. We know that monozygotic twins have a higher concordance than dizygotic twins, and in family-based studies, we know that there's a clear, distinct, and strong heritability of obesity. However, if we look at where on your body you start to accumulate fat, you will see a distinct pattern, and this is something I started to explore as my postdoc in Oxford. And if you look at that, there are monogenic models of this called the lipodystrophies, where you have single gene mutations. You will have selective loss of fat subcutaneously, peripherally, but a selective preponderance to aggregate fat visually, and you will have a cardiometabolic adverse profile. We know that monozygotic twins for fat distribution have a higher concordance than dizygotic twins, and we have a heritability in family studies that is distinct of that from overall obesity. So I was discussing with one of the editors from Nature this morning this slide, and I'm particularly excited about it because it actually represents 37 years of postdoc work. So if you think that GWAS is not big data, I would urge you to reconsider. And for anyone, I see Inga here on the front row, who has spent a lot of their time doing GWAS throughout their career, you will know that it's an enormous amount of work sort of pledged into that. So it started with the first gene discovery, where we raced during the Christmas of 2007 to find the first ever associated variant, common variant with common forms of obesity, which led to a publication in Science of the FTO gene. And 6, I think, now subsequent rounds of GWASs, which I won't talk through because that will be an enormous amount of slide, but this is the culmination and the aggregation of all of those rounds, where we have taken the giant data and we have married it with the UK Biobank data, and talking about selfless, exciting sharing of data in such a way that we can all sort of accelerate scientific discovery. I think UK Biobank is the pinnacle of that. So in this case, you're looking at 27 million variants. It's UK Biobank Plus Giant, and we have about 3,200 associations for overall obesity and about 450 fat distribution specific common variants. So you will see that I've plotted, I don't know how I get the, no? If I want the pointer, do I do, no? You will see the blue upper sort of panel is the female-specific associations, and on the lower red panel, you will see the male-specific panels. And I'm getting, oh, very technical, thank you. You will see the blue panel exemplifying the female-specific associations with the strength of association indicated on the y-axis, and on the x-axis are the chromosomes from 1 till 22. We have a shameful omission of the sex chromosomes, which we still haven't managed to tackle properly within the consortia, but I know Tara Turikainen and Helsinki is doing amazing work on this. On the lower axis, it's a similar plot, but instead the strength of association goes downwards, and it's the association amongst men. So one of the key findings in this was that when we looked at fat distribution, we saw that there is a distinct signature of sexual dimorphism in fat distribution. And when we had lunch today, Hans said to me, but we knew that. Yes, we did know that on a phenotypic level, but we did not know that there was a common genetic underpinning of this, and that it's so distinct for that of fat distribution, where if you look at overall obesity, less than 1% of the loci show any signature of sexual dimorphism. Interestingly, also, as we all know, genetic information can be used for risk assessment. So Amit Kera, together with a group in Bristol, did work where they looked at high polygenic risk carriers and looked how it was associated with extreme obesity and surgery. So in other words, the question was, can we predict who will get extremely obese when they get older by looking at a cross-sectional polygenic risk score? And you can to a certain extent, and this tracks over time, as you can see on the panel here. Following over 25-year, the top decile, about 15% of individuals are in the highest score of polygenic risk and also have the highest phenotype, whereas if you're in the bottom decile, you will not get obese. So Duncan and Nick and I, we had a look in UK Biobank, and it's really important, I think, when we talk about polygenic risk scores to point out that in some cases, blue here, you're in the corresponding tail of a polygenic risk with your phenotype, but in the yellow, you're actually in the opposite risk, which means that you can be in a really high risk polygenic risk, but be of low phenotype, and you can be in really low polygenic risk, but have a high phenotype. So for those of us who are excited about rare variants, you will know that we can now think that there might be single mutation driver that pushes you into one of the opposite carriers, and it's something we're looking at in the lab. Polygenic risk score for obesity is causally associated with cardiometabolic quantitative traits, which is exciting because it gives us leverage in thinking about causality in various pathways. And when you take the GWAS data and put it into a slew of different pathway analyses, this is my dear friend Tune Pash, who together with Lou DeFranke and Joel Hirschhorn developed a framework that they call DEPICT. So what you basically do is you take your statistical summary analyses from your GWAS. The program doesn't know what trait you're looking at. You put in the summary statistics and you get three things out. So the first one is, if you look at the nearest gene of the association, which pathways do you have an enrichment of association in? The second one is the closest or nearest or most likely gene in the associated interval. And the third one is looking at publicly available expression data. Where are the closest gene to the association expressed up and above the background? So I'm just going to draw your eye to, if I can point, this big hairball here. I'm a little bit sort of nervous always when I show a hairball because they're always hard to explain, but I will draw your attention to the fact that almost all of the hairball, which is one of the strongest associations now, lie in adipose biology and also insulin resistance biology. You're thinking fat distribution, we sort of knew that, but when we started to think about fat distribution and we used waist-hip ratio, especially waist-hip ratio adjusted for BMI, it had a very adverse reaction about many of our clinical colleagues who told us, you're just going to pick up skeletal structures, you're just going to pick up muscle, and you're just going to pick up the hip regions. Anyone who is statistically inclined knows that if you use a ratio, and especially a ratio and a just out sort of correlated covariate, it can introduce biases and problems. So we've been very careful in thinking about this. We do see some skeletal sort of structure and some muscle, but mainly liver. We see female infertility as one of the strongest sort of pathways here as well. And Jim very eloquently talked about the development from mesodermal stem cells into various sort of tissue types. And for those of us who are excited about developmental biology, you will know that adipocytes have a common developmental pathway with both osteoblasts and myocytes. So it can be an underlying developmental pathway as well. If we then go in and look at where the genes are expressed up and above the background, for fat distribution, the only cells and tissues that come up are those of adipocytes and adipose tissue, further sort of honing in on which targets, pathways, and target tissues we want to work in. This is Rebecca Feiner. She, together with Joel and Tune, refined this. And if you look at rare variants and push in DEPICT, you get an indication of lipid biology and lipolysis coming up as well, further refining where we want to go with the functional data. In contrast, if you look at overall obesity, again, please note that the only thing we're doing is swapping the summary statistics here. There's an enormous hairball, and in the middle here is the polyphagia, which is the overeating. And all of this is a sort of brain cluster with particular emphasis of neuroendocrine pathways, completely distinct of that of fat distribution. And most people looking at obesity would think that obesity has a very strong adipose tissue component, but we don't see that here. And again, if you look at the same sort of background data going into the system, you will see that there is a strong sort of brain enrichment, but none of other tissues for that of overall obesity. So there's been this sort of ongoing debate about the missing heritability, which you can or cannot be excited about. That's sort of up for discussion, I think. But we have been excited about thinking. We have been really good at looking at the common variants in ever larger sample sizes. We have had arrays coming up where we can look at low frequency variants, which we have done. Didn't find as much as we have thought about, but very few of us been able to look at the very rare or rare variants. Then the dawn of the exome sequencing came, and again with UK Biobank and other large-scale biobank efforts, and with the everlasting data sharing, which we're so excited about, we have been able to look at that. So colleagues from Regeneron and also one or two other labs have had sort of first access to this and have published gorgeous studies looking at about 600,000 individuals and published the first tranche of associations both with BMI and fat distribution. So Nicholas Bayer, Barney Hill, Fredrik Lassen and Duncan Palmer and my team have looked in UK Biobank, done gene burden analyses for BMI, fat distribution, and body fat. I should mention that this is unpublished data that I'd never presented before. Again, taking a leaf out of Jim's sort of presentation and trying to make it a bit more contemporary and not as stale as it could have been. So I have never talked about it, but here we are. We find 19 genes that are significant. We then went in together with Ilkner and Saskia in our team and decided that we were going to start to look at the functional effects measured in adipocytes. Taking a leaf of what we have learned from the GWAS data, looking at lipid accumulation and glucose uptake, both insulin-stimulated and basal glucose uptake. So for me, I got a lot of criticism when we started to do this. People said, you're a statistical geneticist, you're a mathematician, you shouldn't be doing this, you should leave the functional work to people who know what they're doing. Very blessed to have worked with with Ilkner and Saskia on this, and also together with one of the core faculties at Oxford. So when I design things, I get a bit nervous, so we included a lot of positive controls, genes that have been sort of published in the field before, and then we had a bunch of target genes that we were going to look. We have done a complete CRISPR knockout in 2 different cell lines from fat. And here I'm just showing you that the positive control behaved nicely. This is sort of 6 repeats over a bunch of plates that we have done, and we see a significant decrease in lipid accumulation over PPAR gamma, insulin receptor substrate 1 and 2, and TBC1D4. And again, we were really excited when we saw that 5 of our target genes also show a significant effect across these data on lipid accumulation. 4 of them were increasing and 1 of them was significantly decreased. Again, the positive controls behave nicely when we look at the glucose uptake, both in basal state and insulin-stimulated state, but none of them came up of the other genes when we knocked them out. So just wanted to spend a little moment on SLTM, which is associated with increased BMI in males. Since I'm interested in sexual dimorphism, and I've just told you that less than 1% of the loci for BMI show a sexual dimorphism. If you look at putative loss of function variants, you will see a larger PL05 burden in males. And if you look at the knockout, you will see this beautiful lipid accumulation effect and a trend of glucose uptake, but it's not significant. So Samvida Venkatesh in our group also have looked at early onset analyses, basically looking at Cox proportional hazard modeling, comparing individuals carrying the variants to those who have none. And if you look at the males here, you will see a significantly early onset of obesity compared to women where you don't see this. So in parallel in the lab, we've also been thinking about how we gear up for the future. So we're really interested in systematic and large-scale integration of genetic and functional data using various strategies. Just going to give you a quick vignette here from Craig Glastonbury, who I think is in the audience here, who's worked together with Chris Nellocker and also Emil Jarby from Copenhagen, using whole slide image strategies from tissue histology, and then basically developing a neuronal network classifier that gives you the size of adipocytes in tissue, And if we do that, you can size over 20 million adipocytes in over 1,300 samples in a couple of hours instead of days and days. And one really important lesson for our colleagues here is that we do think that you need to size over 500 cells to have tight confidence intervals and have accurate readouts. And that's magnitudes bigger than most of the papers that are being put out today. We know that this work has put forward that the adipocyte area differs across the various depots. Adipocyte area is sexually dimorphic and it's a heritable trait. So we're currently running the first large GWAS of this in an extended sample size where we have more than doubled our sample size in the lab at the moment. I will, however, say that for those of you who think that there are very quick fixes to go about this in smaller sample sizes, you will be sort of disappointed. But we do think we have one or two findings that we're going to try to replicate. In parallel, I have a fantastic collaboration with Melina Klausnitzer, where Melina, together with colleagues from Broad, have sort of come up with this sort of cellular GWAS in a dish strategy. Many people are working on that. We're not claiming that they've done it, but she's doing it together with Hans Hubner's lab, looking at adipocyte-derived mesenchymal stem cells from visual and subcutaneous biopsies, cultivating them, filming them, taking photos, RNA sequencing them, attack sequencing them, then doing GWAS of each of the sort of traits that are extracted, and then linking back the cellular traits with disease. This is just an outline of the flow that is being done. And this has been published by Samantha Lauber and Sophie Strobel, and basically going from a range of different sort of colors that you stain different cellular phenotypes with, and it's a modification of Cell Painter which Ann Carpenter's lab has come up with. And if you do that, you will see that these sort of cellular traits are genetic, that you can do GWAS on them, and now we're developing a strategy for how to link it back to data. So I won't go much further now, but I'm going to say that in summary, we've generated an enormous catalog. We're getting close to saturation of the genetic maps for these traits. We've published the first sort of close to saturated map for height earlier this year, and the next wave of obesity papers are in the making. Of robustly associated common and low frequency variants for various aspects of obesity. I think it's really important that we think of traits as a multifaceted sort of readout of human physiology. We have learned global patterns of novel biology by generating this genetic catalog. And I think you can use the genetic tool a little bit like Jim talked about in the rare variant setting as well, where you can actually use the genome as an information scaffold to learn about traits and conditions and sort of bringing that forward. And I think many of us have an eye towards actually thinking about how we can translate these insights into societal and patient-specific impact. So I think we now have a handle on tissue and cell type of importance. I don't think we can say that we have that on a locus-specific level, though. But with thousands of loci at hand, it's going to be really difficult to go into each locus one by one and do thorough functional work on it, because it's just not financially, time-wise feasible. So I worked on the FTO locus with Melina Klausnitzer. The first paper came out close to 13 years ago now, I think. And there were like 8 papers. There is still a debate as what the conclusive functional story behind that is. And if we go through 3,200 of those loci in that way, we're just going to be really slow. So I do think that these frameworks that we put forward in doing it in a larger sort of screening strategy, where we sort of have a genetic scaffold and use modern technologies, whether it's image analysis with computer vision, or whether it's a multi-omics approach, gorgeous protein data has been shown at this conference, we do need to think about different ways of scaling up, making it faster, but making it in a robust, reproducible, and thorough way. The credible set of most of the variants I didn't talk about, but it's most likely to drive each distinct signal, maps predominantly to non-coding sequence. There's been a lot of attention to that. So it is implying that a lot of the effects are mediated largely through gene regulation. Ultimately, it comes down to the gene that is being regulated, though, and I think that we have no way around that. Sequencing efforts are growing. It's really exciting. Somebody said to me at this conference that we now have over 8 million sequenced individuals globally. So I think coming back to the message about data sharing, we have an effort going on called Brava, where we're aggregating and inviting biobanks globally to come in a sort of communal way, share data, and sort of learn how to analyze rare variants in a similar structured way as we have done with the common variant, and Duncan Palmer is leading that effort. So if you're sitting and want to collaborate, please reach out to Duncan. We need to figure out the causal variants, the transcripts, annotations, pathways, and mechanisms. Somebody said to me, we can actually skip that step. Why would we even bother? We can just go like bypass all of that and go from genetic identification into just thinking about how to go to the disease. I personally think that that will be a shortcoming, because if you don't understand the underlying biology, again, which Jim alluded to very beautifully in his talk, I think you will miss a trick, and I think it's going to become trickier to understand how you actually deliver therapeutics then. So I do think we need to discipline ourselves in being curious about the basic science and sort of drive that and try to understand that. For fat distribution, it's been such a joyous project to co-lead, especially with Karen Moki at UCN, who is not one of the most selfless and smart people, but also a person I can't praise enough as an amazing collaborator. We know that fat distribution is regulated largely through adipocytes. We know that the key pathways that are starting to emerge, and our first trickle of data is sort of supporting that. We're going to look at adipogenesis, we're going to look at insulin sensitivity, and we're going to look at lipolysis. There are no good therapeutics for insulin resistance out in the literature today. We have the glitazones, but 6% of them have liver problems. So FDA has been flip-flopping in redacting them and not. So coming up with new strategies in this field is going to be exciting. And as I talked about, the large cellular screens are going to be absolutely imperative here. Ultimately, I think the challenge is upon us now. I think everyone who's in a trainee in this room, I remember my first ESHG when I was a PhD student, and people were doing linkage studies, and I was just bewowed by, there were the first sort of pipetting robots coming up, and we thought that was amazing. You have a smorgasbord of opportunity in front of you, with tools, with high-performance computing, cloud computing, new analytical strategies in machine learning, but also traditional linear methods. You have data sets of a size and sheer magnitude that we couldn't even dream about. We have quite open sharing, so as long as you have a computational setup, it's your creativity, your excitement, and your joy that can drive this. You can literally be in a quite sparsely resourced lab today and do the most breathtaking science, harness that opportunity, and when you choose supervisors, choose people like Jim, who are kind and want you to succeed. So I'm going to stop there. Happy to take any questions. I, again, just want to thank all my co-author, and especially my lab, who is somewhere out here. You rock. Thank you.
For our award lectures, we don't take questions, but Cecilia is always very open to collaborate and to speak to you. So if you'd like to contact, and if you've got questions about her talk or about her work, I'm sure she'll be delighted to answer you. But join us once again in thanking everybody here in the award for Cecilia as the ESHG awardee for 2024.